The BioFlux system is a complete solution for running live cell experiments under controlled shear flow. Many physiological processes take place in the presence of flow. Adding this capability to an in vitro assay provides a critical increase in physiological relevance. The BioFlux system utilizes well plate microfluidic technology to offer flow assays which feature a convenient well plate format, the capability to run up to 96 assays in parallel, simple setup, and low volume fluidics. The BioFlux system is a fully integrated platform for running live cell assays under controlled shear flow. The system is comprised of a well plate microfluidic device for running shear-based experiments, an imaging system, a shear flow control system, and advanced software for data acquisition and analysis. The ability to introduce shear flow to research and drug discovery experiments enhances the physiological relevance of live cell assays. Shear flow can be used in assays for structure-function relations, mutant and knockout screening, functional cell interactions, morphology and cell cycle analyses, as well as drug discovery and optimization. The BioFlux system utilizes microfluidic devices called BioFlux plates, which feature the proprietary well plate microfluidic technology. Each BioFlux plate comes in a pre-sterilized SPS well plate format. BioFlux plates come in a variety of designs and formats, but each works in a similar fashion. Each plate contains an array of micron-scale flow channels which run along the bottom of the plate. The wells of the plate serve as reservoirs for the reagents going into and out of the flow channels. Here we are showing the 48-well high-shear plate. This plate contains 24 experimental flow channels, each connecting to an input well and an output well. Fluidic reagents, such as cells, proteins, and drug compounds, are loaded into the wells using standard pipetters or other liquid handling instruments. Once the wells are loaded, the BioFlux pressure interface is attached to the well plate. This creates a pressure-tight seal around each of the wells on the plate. A user-defined shear flow is entered into the BioFlux software, which creates a precisely regulated air pressure from the BioFlux controller. The controller contains a software-controlled pump, which distributes pressure to the wells of the BioFlux plate. The air pressure is transferred via the pneumatic tubing to the pressure interface. Once the well sees this increased pressure, the reagent will flow through the port at the bottom of the well and along the direction of the flow channel. Once the reagent clears the flow channel, it will begin to pool up in the output well. In this manner, a variety of cell ligand and cell-cell assays can be performed. Prior to the experiment, coatings such as cell adhesion and extracellular matrix protein molecules can be introduced. The coating is introduced into the inlet well and, using pneumatic pressure, flowed through the channel and allowed to incubate. The excess coating is removed and a washing buffer is introduced. The channel is now coated and ready for the rest of the experiment. Cellular monolayers can be formed on the bottom surface to perform cellular interaction studies. Cells in suspension are introduced into the channel and allowed to settle. The cells are allowed to attach and form a tight monolayer under controlled temperature and environmental conditions. At this point, additional circulating cells can be added into the chamber under controlled shear flow. Some BioFlux plates have an additional input well, which allows a second reagent to be introduced, such as a washing buffer, media, or drug compound. These two sources can be dynamically switched to allow for time-resolved addition of reagents or drug compounds. The two input channels can also be added simultaneously to enable applications such as wound healing and chemotaxis. Each experimental channel has a dedicated viewing area, which can be observed on an inverted microscope. BioFlux plates have 180 micrometer glass cover slip bottoms and are compatible with high-resolution microscopy. Images can be acquired in bright field, phase, fluorescence, and confocal modalities. In summary, well plate microfluidic technology provides higher physiological relevance, increased throughput, simple, fast, and reliable setup procedure, and reduced reagent consumption. Image data is collected using the BioFlux imaging system, a high-end inverted microscope that has been adapted for use with the BioFlux system, but can also be used with most commercially available inverted microscopes. The BioFlux imaging system supports bright field, phase, and fluorescence microscopy data acquisition, and comes with a customized automated stage for rapid scanning of BioFlux plates. Data collection is accomplished by the BioFlux Montage software, which provides a complete package for hardware control, data acquisition, and image analysis. This software serves as a single point control for the entire system and includes all the necessary controls for the automated stage, CCD camera, Z-drive, automated fluorescence, and BioFlux controller. Data analysis is automated with innovative modules which couple directly to the BioFlux flow control. The Montage package offers sophisticated image analysis capabilities, as well as customized modules designed specifically for analyzing BioFlux applications. A wide variety of experiments can be performed on the BioFlux system. 3 representative examples are presented here to illustrate the versatility of the platform. The first example is a platelet aggregation study. The BioFlux channels are coated with collagen 1, and whole blood is introduced under controlled shear flow. Whole blood without drug compounds is used as a control, which shows thrombus formation on the collagen-coated channel. Experimental channels contain whole blood pre-mixed with 20 micrograms per milliliter of anti-GP2B3A antibody inhibitor, which prevents platelet aggregation. The area coverage of platelet adhesion is then calculated using the Montage software, which quantitates the effect of the drug compound. Less than 1 milliliter of blood is needed for each experiment, which enables more experiments per each blood draw versus conventional flow cells. The second example is a leukocyte adhesion experiment. The BioFlux flow channels are coated with a cell adhesion molecule, and isolated leukocytes are pre-mixed with a panel of VLA4 antibodies in varying concentrations to study the effect of the compounds on cell adhesion. The leukocytes are introduced into the ICAM-coated channels at a shear flow of 0.8 dyne per centimeter squared for 10 minutes, then the unbound cells are washed away. The Montage software is used to calculate the number of adhered cells, which quantitates the effect of the compound. The software also generates critical cell morphology data, such as shape and size. Up to 96 cell adhesion assays can be run in parallel using the BioFlux system. Biofilm assays are a third example of experiments that can be performed with the BioFlux system. Here, a panel of antimicrobial drug compounds is introduced to a bacterial biofilm grown under shear flow in the BioFlux system. Live versus dead cells are stained at an endpoint to assess the efficacy of the antimicrobial compounds. BioFlux Montage is used to quantitate compound efficacy based on fluorescent stain intensity. In conclusion, the BioFlux system enables a diverse range of live cell experiments to be performed under controlled shear flow. This brings a higher physiological relevance to research and drug discovery assays and provides workflow benefits such as higher throughput, easier setup, and lower volume requirements. Please visit www.fluxionbio.com for more information on the BioFlux system or call to speak with a product representative.